Okay, so good morning, everyone. And today we are going to deal with this software that is used for modeling biochemical systems. Okay, and this software is called as Copasi. Okay, so please keep your mic on silent. Huh? So that if you want to share anything, then only unmute yourself. Okay, huh. So I would be kind of taking you on a tour on this software, how to build up an elementary biochemical system, and how do we simulate our biochemical system. So far so good, you people. I hope you might have faithfully written the codes of the molecular system that I have already told you to write. Okay, about simulating the biochemical system of, say, some reaction, and simulating the effect of inhibitor by finding out the expression for competitive and different type of inhibition that we know. And the final program that I have asked you to write about was the biochemical pathway A to B to C, and I have told you on plotting what is the effect of what is the effect on concentration of metabolite C. I now hope that you have done those things faithfully, and today we will be dealing with how how softwares are available, which softwares are available that can be used for doing such simulation and more complex task. Okay, so the thing is that let me start out to share my screen. So I really hope that you people have already downloaded and installed the latest version of Copasi. Okay, so besides downloading, there are a few things that I think you people should know. Now, do you see my screen?
Yes, sir.
Okay.
Yes, sir.
So I hope कि आप यही से डाउनलोड किया होगा, right? This same website. Okay. Everyone has downloaded an appropriate version from this website, right?
Yes, sir.
Okay. So I would like to share a few things about this website, and what are the features that you can explore right from this website before we start out on directly working on that software. There are a few things that I think you should know about the website first. Okay, so this is the home page, and on this home page you might have used this download option to download the latest version of Copasi as per your operating system. Okay, so there is also another tab called as research tab over here. Okay, and this research tab will it kind of keep tracks of how many papers are published that cites this Copasi as such. Okay, so I would advise you that you should always check out like what are the recent publications that are being that have used the software in their research. Okay, and how they have used it. So it will give you a kind of idea on whatever I am teaching now, what are its application. Okay, how creatively people have used this software. Okay, what kind of systems that these guys have developed, what kind of results they get, how do they interpreted those results, what conclusions that they have drawn from whatever data that they have generated. So this is very important in order to for you to appreciate what this tool can do in biological sciences. Okay, so it is a very big list of year-wise list of publication. Okay, so I would advise you to go through some of the interesting papers that are present over here. So it is very important, right? Then you have got this support, in which support tab bhi hai, in which you have got this video tutorials. Okay, and these people also have got their own YouTube channel. So I would advise you to go to this YouTube channel and check out these videos that demonstrates how you can set up the simulation, or how you can use different utilities from this software. Okay, so this is where you can get some extra help if you wish. Okay, then there is a user forum. Okay, using this user forum, well, this user forum is also quite active. Okay, all you need to do is to do a registration over here. Okay, and well, you can talk to other people who are working over here. Okay, you can post your questions, and maybe you can get answers, right? So I would advise you that you can go to this user and use this user forum as well. Most important thing is getting known to your software, right? So there is a complete online manual of the current version that is released. Okay, so I would advise you to go through this user model. Okay, and this user guide, so that you should spend some time on knowing the software before directly using it. Okay, so I would advise you to go through what are the using I mean, reading the user manual would give you an idea on what premise this software is built on, what it can do, what it cannot do. Okay, what are the features that are somehow not very apparent when you start to use it, unless and until you dig out into more details, right? So all these things can be gained by reading out simply your user manual. So whenever you use any software, not only Copasi, right? It can be GrowMax, it can be NMD, it can be Autodoc, it can be anything. So always make sure that you at least go through its user manual first. Okay, and just in order to be sure what are the capabilities that of this software, and where and how it can be used, that would enable you to have that insight. That is why you should go through this user manual. Okay, so there are other how to install it and history of tracker and everything, but what I wanted to convey today is that go through these video tutorials, register yourself on these user forums, ask questions there. If you are facing, people would answer it for sure. Get known to the software by reading the entire user manual. Okay, then of course you have got links to a bigger project like Jigsaw Run Manager. Okay, and multi-state model builder. Well, these are advanced or higher level of projects that can be used to simulate cells, and say for example systems that are of much higher uh, complexity in nature. Okay, so we are not going to deal with these things for you at this current elementary stages, but just for you to know कि यहाँ पर है. Okay, then if there are events, workshop वगैरह कहीं पर होगा, tutorials होंगे, तो just go and check it out. Okay. Then about teams and funding, it is not necessary that you go over here. Okay. So I really hope now you have downloaded, you have installed this software, and from today's discussion I would request you, advise you to explore all these things that I have mentioned today. It will only help you. Okay, it is not mandatory to do that, but if you do that, it would be helpful for you in order to understand and appreciate this software. Okay. This is what I was talking about. Okay, sure. Now let us talk about our software. Okay, now again I will share the software that we are going to run. Okay, so when you click on the after installation, I hope you have already done it. So when you install this software and when you double click on its icon that might appear on the desktop, you might open up with this window first. Okay, so now my primary objective of today's assignment or today's practical is to make you aware of this software. Okay, take you around and show you some functionalities, try to explain what these features are. Maybe I will also by doing so we would also create a biochemical system, try to simulate it. Okay, and this would be a very basic practical. Okay, and the objective is to let you know where things are and how to use them. Okay, so let us begin now. This particular software is my screen available? Can you see this screen?
Yeah, sir.
Okay, दिख रहा है सबको?
Yeah.
Okay. So as you can see, most of the time in my further or subsequent lecture, or you can say this practical demo, I would be talking about left hand side and right hand side. So when you open up this software, what you would prominently notice is that it is being divided into two panels, the left hand panel and the right hand panel. Okay, so when I say subsequently that do things in right hand panel, that means you are supposed to look out somewhere over here. And when I say that do something in right-handed panel, left-handed panel, you are supposed to check out somewhere over here. This is RHS and this is LHS. Okay, left-hand side and right-hand side. So as usual, this is a Windows-based software, so it will have its typical menu like file that would contain everything about opening and saving and exporting and importing, right, and closing and quitting. Those typical menu are present over here. Okay, if you want to start out with a new project, if you want to open up already saved project, if you want to import anything in SBML format, which we are going to talk about a bit later in after a few practicals. Okay, so these basic commands of opening, saving, initiating, closing, all of those are over here in your file menu. You have got edit menu, in which of course you can do copy, undo, paste, undo history and everything. Then you have got these tools which has got different commands. Okay, so we are not going to talk about these things in this particular session. Okay, maybe we will use some of its components in advanced stages. Okay, then there are windows, so you can hide the main toolbar or show the main toolbar, help के लिए how do we set up a simple wizards, if there is an update checker, okay, about the team that has developed this Copasi license and everything. Okay, so well, these are common menus that are usually present in any GUI. Those are that are Windows based softwares, right? So initially, when this Copasi was published long back ago, when we myself and Dr. Vivek started using this software long time ago, at that time we didn't had this GUI, right? And we only had the command line version, and it was difficult at that time for writing all those script, making sure everything is there in the right place, and then running it from command line. It was a difficult and tedious task. But now with the advancement of this graphical user interface, things have become quite lot easier to perform such biochemical simulations. Okay, so you guys must be very lucky, so that you now need to only work on this GUI, so that you don't have to worry about writing all those scripts and coding and everything. Okay, so it was difficult in the past times. Now it has become quite easier. Okay, so now let me introduce you what are the things that are over here on the left hand side. Okay, as you can see, there is a uh, you know uh, a drop down menu called as Copasi over here, which if you try to use this triangle, if you open it up, it gives you at least 5 menus over here, right? Model, task, output, specification, functions, and units. Okay, so one by one I will let you know what are the utilities that are present in these submenus as well. We would be using a few of them. Okay, and appropriately, if you by default, the model menus first page is shown on left hand side and right hand side as well. Okay, so if you kind of open this model menu, you can see then there are two more sub menus over here: biochemical and mathematical, as well as diagrams. Okay, so ये जो biochemical है, this is one of the most important menu in which you people are going to work for most of the time today and in subsequent few sessions as well, right? So in this biochemical menu, it enables you to generate a biochemical model, and in order to generate a biochemical model, you need to define a few things. Okay, and you can say that these are preparatory stuff. This biochemical menu will uh, help you to set up and get running with the model that you are trying to work with. Okay, so there are different submenus over here, like you can define compartment, you can define species, you can define reactions, you can define these global quantities, what are the events that your model is going to experience. You can also save things, you can also view what are the different parameters that you have set up in your model, and you can save the parameters so that you can use those parameters up and ready if you are trying to build up a new model, or if you want to start out with the previously saved model. तो उसके लिए यहाँ पर parameter overview काम में आता है. Then you can set the parameters. So well, the thing is that uh, this bracket में जो zero दिख रहा है यहाँ पर, that would indicate the number of things that are present in this submenu. So now you see over here that in almost all of the submenus, the value is 0. That means we haven't defined anything. We haven't defined a compartment. In that compartment, we have not defined species yet. We have not defined in that compartment how many species are there, how many species are undergoing what kind of reaction. Therefore, everything is set to 0. Okay. So let me tell you everything about this biochemical menu first. Okay, and then we would be talking about this mathematical diagrams and task one after another as we go through our tutorial today. Okay, so let us talk about this model first. So the thing is, whatever you would work in Copasi, you would work in a given model. Okay, and on that model you would be performing task. Okay, so the first and foremost thing would be to set up a model, and once you set up this model, the next subsequent and logical task would be to perform some analysis, to perform some task on this model. So first let us try and understand what this model is all about and how do we set up it. Okay, so when you click on this model, on the right hand side, as you can see over here on the right hand side, you will have this model. Okay, the name of this model. Okay, so you can rename this model to, maybe I will rename it to test. Okay, since we are using a test model. Okay, and then in this test model on right hand side there are three important tabs. Okay, so the very first and default tab that is open for any model is about the details tab. ओके, तो ये डिटेल्स में क्या क्या है आपको यहाँ पर अलग अलग फंक्शनैलिटीज दिखाई देंगे दैट विल गिव यू व्हाट आर द डिफॉल्ट units that are considered in this model. Okay, when you try to simulate some model, okay, when you try to simulate some model, the default time that will be used for simulation is given in seconds. Okay, so quantity unit of different species that you are going to use would be in molar. The volume of the compartment that you would be using would be in liters, right? Unit area would be in terms of meter square, length would be in terms of meter. Okay, and you also have got something called as Avogadro's number. Okay, and there is also something called as apply stochastic correlation to rate laws. So let me talk about something about this stochastic and everything. Okay, तो यहाँ पर this apply stochastic correction to rate laws, it is checked. Okay, so what you need to do for time being is that we won't require this stochastic correction to our rate laws. Okay, now to talk about this why we are unchecking it. Okay, so simulations can be either deterministic or stochastic. Okay, तो ये चीज थ्योरी में डिस्कस हुई कि आपकी डिटरमिनिस्टिक सॉल्यूशंस एंड स्टोकेस्टिक सिमुलेशंस. Okay, I think Vivek might have told you. So whatever model that you would generate, that can be either deterministic or it can be either stochastic. So what are the properties of deterministic model is that the output of the model is fully determined by the parameter values and the initial conditions as well as the parameters that are used. Okay, while on the other hand, if you try to use the stochastic model, it would possess some inherent randomness. Okay, and due to the randomness that is involved in your model, the same set of initial parameter values and initial condition might lead you to give an ensemble of different outputs. Okay, so what I want to convey is कि if you are using deterministic model, that would mean that the output of your model would be completely dependent on what are the initial conditions as well as the initial parameters of your model. Okay, if you remember in the very first practical, I have kind of briefly describe what are parameters of a biochemical model. Okay, if you are kind of lost, then maybe after this lecture you can go and check out my first video of this practical sessions. Okay, तो for time being सिर्फ इतना ही ध्यान में रखो, अगर डिटर्मिनिस्टिक मॉडल आप बना रहे हैं इसका मतलब क्या है, the output of the model would be completely dependent on the parameters that you are stating and the initial conditions of your model. On the other hand, if you are using the stochastic model, stochastic है इसका मतलब क्या, there is the possibility of inherent randomness. You are trying to you are trying to simulate randomness in your system. ठीक है, and therefore due to randomness uh, randomness की वजह से क्या होगा, the same set of parameter values as well as if you give the same set of initial condition, you might obtain some varying results. Okay, for time being we are using only deterministic value. Okay, deterministic model as such. Okay, so यहाँ पर आप initial time भी set कर सकते हैं. Okay, so initial time is set to 0 over here. Why? Because we are going to start out our model right from scratch. Right, इसके पहले अपन ने कुछ काम किया नहीं है. Right, well, but say for example, if you have if you have run the time course simulation and or you have run these time course simulation, or if you have performed steady state analysis, then you can go for further analysis like metabolic control analysis, flux balance analysis, in which you can use the model from, well, say for example, the model that has already obtained the steady state. So at that time you can state the initial time. Well, say for example, if you have performed a time course simulation or a steady state analysis of say some 1,000 seconds, then when you perform metabolic control analysis or subsequent flux balance analysis, you can start out at the time where your time course simulation has finished. So these things can be set over here. Okay, so this was about your details tab. इसके अलावा यहाँ पर एक annotation tab भी है, in which what you can do is that you can provide annotation about your model. Okay, like when it was created, who are the authors that are present in this project. Okay, so these things would eventually be helpful when you try to submit your model in say some public database. Okay, so all the essential values that would be required to submit your model in any submission system, like बायोमॉडल नाम का एक बहुत बड़ा डेटाबेस है जिसमें आप मॉडल्स ज्यादातर ये सबमिट करते हैं, तो जो ऑथर वगैरह का नाम है वो यहाँ से लिया जाएगा. You can cite over here what are the references that you have used for obtaining kinetic parameters and initial conditions or initial concentration of your metabolite, anything. Okay, you can provide a description of your model, at what time or you can say how many times this model was modified. Okay, so you have got lot of things to tell about your model, so you can do it over here. Then the next tab over here in your model section is about RDF. Okay, तो ये RDF browser जो है, well, that is somehow dealt or that is present in connection with SBML format. Okay, we'll come to this thing later when we try to import and export this model. Okay, for time being you can only consider कि भाई ये जो RDF browser है that is related to input and output and something related to SBML format. Okay, so now I have already told you that this is the model that you are dealing with. Okay, then let us discuss the first submenu in your software, that is biochemical. So this submenu is generally used to set up your system, as I have already told you. Okay, तो इसमें सबसे पहला मेनू कौन सा है, something called as compartment. Okay, so this particular, if you click over here, you would be taken to its own subsequent menu on the right hand side. Okay, बायोकेमिकल था जब अलग था, कंपार्टमेंट है जब अलग है, स्पीशीज है जब अलग रहेगा. Each and every sub menu on left hand side has its corresponding menu on the right hand side. So when you are over here, if you double click over here, you would be looking for, how do we set up the compartment. You need to state it over here. Okay, so we would, if there are many compartments, then you can search over here. Okay, so the thing is that everything that happens in your model would happen in, say, some compartment. So we can make a name of compartment, like if you are working on, say, metabolic pathway that involves two compartments. Some of the reactions happen in mitochondria while some of the reactions happen in cytosol. So there would be two compartments. Okay, one of the compartment name would be mitochondria, another name of the compartment would be cytosol. You might have Golgi complexes. However number of compartment you want, you can state it over here. Okay, so for time being, we would be using a simple compartment. We would call it as cell, because whatever biochemical reaction that we are going to simulate, well, we would assume that it is happening in cell. Well, let us not go into more details of in cell exactly kahan par ho raha hai. Let us consider that it is a simple prokaryotic cell. Okay, good. So since we don't have anything, it won't show it over here. You would name the compartment as cell. तो जैसे ही cell बताता है, the number of compartment would be updated over here. Although you have got a new compartment over here, it is not considered as the compartment that is to be used in your subsequent simulation. Okay, so we have got cell as a compartment. Now this cell is associated with certain attributes that are shown over here. उसके आगे जो कुछ भी attribute है, well, those are all attribute of your cell that can be changed. Okay, so the very first attribute of your compartment, which we have named it as cell, is the type. Okay, so the types, this attribute type is usually used to define size of your compartment. Okay, whether its size is, now you can see that if you click over here, you will get a drop down menu, and there are three values, or you can see there are three attributes that this compartment can possess. Okay, so cell can be either fixed, your cell can be assign your cell can have either assignment type or it can have ODE type. So when we say, or when we select a fixed type to your cell, that would mean let's say, for example, it would mean that uh, by keeping it fixed, it would be like stating that our cell is not growing. Okay, and have fixed volume, where initial size, okay, we have where initial size is one unit, that is 1 liter. Well, now it is quite very illogical to state कि अपना cell 1 liter का रहेगा, but this is how the minimum size of compartment for this software is. But just consider that you have a cell that has got unit size of 1 liter. Over here, the rate is 0. ठीक है, rate is at present 0 because of two reasons. Mainly, we have not done any simulation yet, बरोबर है? Therefore everything is at its initial stage. And second thing is that we have already used it as a fixed type. Okay, we have already assigned it as a फिक्स टाइप, इसकी वजह से यहाँ पर क्या है, इसका रेट 0 है. इट माइट चेंज इन ड्यू कोर्स ऑफ टाइम. ओके, तो इसके अलावा यू हैव गॉट टू मोर types that can be attributed to yourself, what we call it as assignment type or what we call it as ODE type. Okay, so ODE stand for ordinary differential equation. Okay, so if you assign it ODE will change with time over the course of simulation. Okay, if you use assignment type, okay, that would mean ki if we select either ODE or if we select ODE or assignment type, that would mean that you would have to provide certain expressions that are related to either assignment or either ordinary differential equation. Okay, so when we say that we are assigning a cell type, that would mean that there must be some expression that is assigned to your cell. Okay, बात समझ में आ रही है आपको? If we select assignment type or ODA type, then we will have to give expression in this initial expression or expression columns. Okay, so that we can either assign this particular compartment with some assignment, some expression or say some ordinary differential equation. वो अपने को यहाँ पर बताना पड़ेगा. ठीक है? Now you know, the moment I change fixed type to assignment type, you might have noticed कि यहाँ पर देखो ये marks आ गए, right? ये यहाँ पर marks आ गए. Well, these are what we call it as exclamatory marks, or you can say जहाँ पर भी ऐसे mark आ गए समझ जाने का कि कहीं पर तो भी गड़बड़ है. Something is going wrong over here. Okay, so now in order to check what is going wrong, you know, it says कि मॉडल में प्रॉब्लम है. मॉडल में एग्जैक्टली कहाँ पर प्रॉब्लम है, उसके कंपार्टमेंट में प्रॉब्लम है. कंपार्टमेंट में एग्जैक्टली कहाँ पर प्रॉब्लम है, उसके सेल में प्रॉब्लम है. राइट, सेल में प्रॉब्लम कहाँ पे है, बिकॉज वी हैव नॉट असाइन एनी एक्सप्रेशन टू ओवर हियर. ठीक है, वो बताता है कि यहाँ पर प्रॉब्लम है. यू नीड टू सेट सम एक्सप्रेशन ओवर हियर. ओके, so always before you proceed, make sure that there are no any warning symbols there in your model. तो यहाँ पर warning symbol क्यों आया, because in compartment session we have used assignment as its type and we have not yet provided their expression to whom that cell is to be assigned with, right? So we would again change it back to fixed type so that the warning signs go away. Okay, बात समझ में आ रही है आपको ये? For time being we would be using only fixed type. Okay, so you see, the moment we change to fix, all the warnings are gone now. Okay, so this is one of the lesson. Do not proceed further unless and until you make sure that there are no errors. And this way the software has become more intuitive. Okay, so that it will tell you that something is wrong. So such thing was not possible when you use your command line version, right? So you have to yourself figure out कि कहाँ पर गड़बड़ हुई है. Otherwise the script will not run at all, right? So over here at least this GUI tells you कि where things are going wrong, and you should always keep track of those things before you proceed further. Okay, so now the bottom line is that we have defined the compartment in which our metabolic pathways are going to be functional. Okay, so now let us talk about this species. Okay, so this menu and the subsequent table that you get on your right hand side are used to declare the species that are going to be present in your cell and the species that are going to be part of the reactions that we are going to subsequently set up. Okay, so in this species table we have separate columns, as you can see. Okay, you can give define the name of the species in this particular column. Okay, well, let us say that we are having species 1, metabolites name S1. Okay, we are having this metabolite S1 species 1 in the compartment cell. Of course, you can also define attributes to this species. Okay, you can either have reaction, okay, fixed, assignment, ODE. Okay, fixed is well, say for example, if you set this attribute to reaction, that would mean that the concentration will change by participation in a reaction in course of simulation. Okay, तो अगर आपने ये स्पीशीज का एट्रीब्यूट रिएक्शन टाइप रखा तो इसका मतलब क्या होगा? वेल, इट विल टेल यू कि हाउ द concentration of this species will change in the given simulation. Okay, so if you set it to reaction, that would mean that the concentration of this species S1 will change by participating in certain reaction in the due course of simulation that we are going to set. If you assign it fixed, that would mean the concentration will remain fixed and do not change in the simulation. Okay, if you give this attribute as an assignment, that would mean the concentration of this particular species will change as per the expression that you are going to assign in these two columns, either expression column or initial expression column. Okay, so it is also possible that you can change the concentration of say certain species as per the given assignment. Okay, well, these are very specialized stuff that you might do at higher level of simulation when you are quite comfortable, right? ODE यानी क्या? The concentration will change as per define ordinary differential equation, which you would of course have to define in this two columns, either of the two columns. Okay, so all in all, defining the types would eventually define or it would signify how the concentration of the particular species in your compartment will behave or change over the course of time. Okay, for time being, we would keep the species to the reaction type. अपन reaction type define कर रहे हैं यानी क्या? This defining this attribute to reaction would mean that in due course of simulation, the concentration will change by participation in the reaction in the course of subsequent simulation time. Okay, okay, so ठीक है, this is what it is. Okay, now you can also change the initial concentration. Okay, for time being it is only one. Okay, the initial concentration has to be set up for species using this initial concentration column over here, right? Concentration tab is can be set is 0 because देखो अभी initial concentration कितना है? 1 है, right? It is the default value. Okay, so we would kind of change this initial concentration in due course of simulation. Okay, ठीक है. So now let us talk about this reaction over here. Okay, तो अभी क्या करेंगे? We would keep two species over here. Okay, species 1, new species would be species 2. Why? Because we are having two species, so that species 1 would get converted to species 2 in our reactions. Okay, and we would set the species 2 to 0. Okay, why? Because species 1 after reaction would create species 2, therefore the initial concentration must be 0. right?
Yes, sir.
Okay. So this is all about, we have now two species, species 1 and species 2 in our model, which has got one compartment called as cell. Okay. So okay. तो ये जो चीजें है यहाँ पर इनिशियल एक्सप्रेशन और वेल आई हैव ऑलरेडी मेंशन दिस थिंग. ओके, नाउ यू आल्सो हैव समथिंग कॉल्ड एज नॉइज एक्सप्रेशन. ओके, जो पहले भी था, कंपार्टमेंट में भी था. सो व्हाट एग्जैक्टली इज दिस नॉइज एक्सप्रेशन टैब? सो व्हाट डू इट डू? इसका एग्जैक्टली काम क्या है? well what is to be expected here? Okay, let me explain you in simpler terms. Like sometimes if you have a deterministic simulation, but you want to see the effect of noise. Well, say for example, अगर आपने initially उसको stochastic करके रख दिया है. Okay, we are working in a deterministic simulation, but say for example, if you are working in stochastic model, that would mean कि उसमें randomness रहेगा. Randomness रहेगा इसका मतलब क्या है कि your data would have any it is it is possible that you might be expressing noise in your data because system can be chaotic. Okay, and in such case you might need this noise column. Okay, so in order to keep tab of what is the randomness or disorderness in your system, what is the level of randomness or disorderness in your system, or you can say how chaotic is your system. So let it be. If you for time being, if you are not able to grasp it, well, we won't go in more details of it for time being, but for time being you can only say ki, okay, this is used in case of uh, stochastic simulation, and it would take care of randomness. Okay, for time being इतना भी ध्यान में रहा तो भी ठीक है. ठीक है. So this is what we have done. We have got two species. Okay, and I have explained you what is this all about. Okay, good. Good. सबको समझ में आ रहा है बेटा क्या हो रहा है यहाँ पर?
Yes, sir. Sir, noise expression like a question था.
हाँ, बोल.
So we can manually input that, right?
नहीं, वी कैन
नहीं.
कीप ट्रैक ऑफ दोस थिंग्स कि भाई कितना नॉइस है ये S1 के डेटा में आगे चल.
Okay. अच्छा, so after simulation we can get an idea of that,
गेट
right?
एन आइडिया. हाँ.
Okay, got it.
Okay. और मे बी यू
Okay,
कैन
sir.
ऑल्सो कंट्रोल इट, राइट? हो
Right.
सकता है. Well, let us not go over there. ठीक है. Our my idea is to keep things simple. मैं जितना ज्यादा डिटेल में जाऊंगा things would appear कि ये बहुत complicated है. That is not what my objective is.
Okay, sir. Okay.
So now our left hand side species tab में अगर देखेंगे तो कितने दिख रहे हैं आपको? यहाँ पर दो स्पीशीज दिख रहे हैं, S1 and S2. ओके, तो हर एक स्पीशीज का एनोटेशन. ओके, अगर आप इसके ऊपर में क्लिक करेंगे, ठीक है, तो यू कैन ऑल्सो गिव डिटेल्स ऑफ दिस स्पीशीज. बात समझ में आ रही है क्या आपको? मैंने कहां पर गया? नाउ आई हैव क्लिक्ड ऑन द लेफ्ट हैंड साइड. तो आप ये स्पीशीज का भी एनोटेशन दे सकते हैं, डिटेल्स दे सकते हैं, राइट? व्हाट इज द इनिशियल कॉन्सेंट्रेशन ऑफ दिस स्पीशीज ओवर हियर? जो डिफॉल्ट अपन ने 1 रखा था, डू यू रिमेंबर? स्पीशीज 2 का डिफॉल्ट अपन ने 0 रखा था. व्हाई? Because initially when we begin our simulation there would be only reactant, there won't be product. Species 2 is our product, therefore uska initial concentration 0 hai. Okay, then you can it tells ki ye kaun se compartment mein hai, what is the type or say for example attribute that you have already assigned to this species. You can also change it over here. Okay, initial concentration kya hai iska, right, where it is involved. You can also provide notes over here telling ki what that species is. If you're kind of if you are planning to modulate, or if you are planning to generate a system in which there is a very big metabolic pathway and you want to keep tab of what these species do, तो उसके लिए आप उसका नोट्स बना सकते हैं, उसका आप एनोटेशन भी दे सकते हैं, राइट? कि if you have obtained the initial concentration from say some paper, then you can refer that paper over here. If someone gave you the initial concentration, you can put the author's contribution over here, you can describe its relationship, right? Okay, so what property do these species have? Okay, if it is related to certain resources, okay, तो वो आप भी सिलेक्ट कर सकते कि ये कहाँ से लिया है अपन ने, राइट? व्हेन इट वाज मॉडिफाइड, ओके, तो ये डिटेल्ड एनोटेशन आप इसमें डाल सकते हैं, एंड these annotations would also be get absorbed into your final model when you submit it to say some database. ओके, तो जो कुछ भी राइट हैंड साइड में है हर एक का डिटेल्स, नोट्स, एनोटेशन एंड आरडीएफ, ये सारी चीजें रहने वाली है. ओके, इज इट क्लियर?
Yes, sir.
ओके, सो नाउ लेट अस मूव ऑन. तो अभी अपन ने सेल डिफाइन कर लिया, अपने सेल में रहने वाले जो रिएक्शंस है उसके रिएक्टेंट एज वेल एज प्रोडक्ट डिफाइन कर लिए, राइट? नाउ इट इज टाइम फॉर अस टू डिफाइन द रिएक्शन. ओके, सो ये जो है वेल आप रिएक्शन पर क्लिक करने के बाद में यू वुड ऑफ कोर्स बी टेकन टू से सम टेबल जो बाकी के जैसे ही है. Okay, so over here in this table, you are supposed to define the reactions that are present in your model or that are present in your system. Okay, so अभी देखते हैं कि यहाँ पर क्या क्या है. Okay, this is over where you are going to define the reaction. Okay, so you need to state in this reaction column what reaction these two species would be undergoing, right? So you can define the reactions over here. Okay, so उसके बाद में क्या है, okay, name of the reaction you can say over here. ठीक है, you can give any name. Okay, so we'll say for example you can say it is you can say phosphofructokinase or anything. Okay, you can give any name over here, right? So for example we would give reaction name R1, right? तो अपन ने एक रिएक्शन डिफाइन की R1. ओके, और जैसी रिएक्शन डिफाइन की इट शोज काइंड ऑफ यू नो some marks over here which tells की गड़बड़ है. Why? Because you have not defined the further stuff, right? तो ये रिएक्शन की भी प्रॉपर्टीज आपको डिफाइन करनी पड़े. सिर्फ आपने रिएक्शन डिफाइन किया, यू हैव ओनली डिक्लेयर्ड द रिएक्शन, बट यू डिडंट टेल ओवर हियर कि यू नो कि ये रिएक्शन क्या करने वाली है. ओके, सो इन ऑर्डर टू चेक आउट, ओके, तो पहले मैं आपको ये टेबल के बारे में बता देता हूँ कि क्या है. ओके, सो ओवर हियर यू आर सपोज टू स्टेट की रिएक्शन कैसे बिहेव कर, मतलब व्हाट आर गोइंग टू बी द सबस्ट्रेट एंड प्रोडक्ट ऑफ योर रिएक्शन. Then over here you are going to tell ki what is the rate law that this reaction is going to follow. Okay, so rate laws आप ये column में define कर सकते हैं. There is also a flux column that displays the flux through the reaction. Okay, this will change during the course of simulation. जैसे जैसे reaction में रिएक्शन आगे बढ़ेंगे उसके अंदर से या तो फ्लक्स इंक्रीज होगा या तो फ्लक्स डिक्रीज होगा. सो दीज थिंग्स कैन बी मॉनिटर्ड ओवर हियर. तो वी हैवंट सिमुलेटेड एनीथिंग, देयरफॉर यहाँ पर उसका वैल्यू 0 है. ओके, नाउ जैसे ही नाउ नोट दैट वी हैव देयर इज समथिंग रॉन्ग ओवर हियर. नाउ लेट अस ट्राई टू फिक्स कि क्या चीज गलत है. ओके, तो इफ यू क्लिक ओवर हियर, ओके, यू वुड सी कि something is highlighted over here in pink. Pink मतलब wrong. Pink means wrong. You need to change something over here in order to make things right. Okay, so it is now trying to tell you कि you are supposed to state the reaction over here, राइट? तो हाउ डू वी डिफाइन ए रिएक्शन, मैं आपको बता देता हूँ कि आई एम सिंपली सेइंग दैट अपना रिएक्टेंट कौन सा है, इन आवर केस इट इज S1. Okay, then I am giving a space, then I am giving a dash over here, and then I am giving greater than mark. So I am kind of creating an arrow over here. Okay, that shows कि this S1 is being converted to S2. This is our reaction. Now things have turned from pink to blue. That means things are okay. So if you click anywhere outside, okay, these things would go away, right? Okay, so the moment you change this reaction, or you define this reaction, so एक और चीज आपको ध्यान में रखने की जरूरत है कि spaces are usually not allowed in the names of this reaction. Okay, so say for example, if you have any reactant, we'll say for example glucose 6 phosphate, and you have named it like glucose 6 and then space P. Okay, it will still show you that there is something wrong over here. In such case, what you can do is that you can use it in double quote, and then things would work properly. Okay, so if there are spaces or special characters or symbols in your name, you should place it in double quote so that it won't recognize it as an error. Okay, so always keep tabs of all these spaces and everything. Okay, बात समझ में आ रही है आपको? Keep tab of spaces. Okay, if there are spaces in species name, then use them in double quote while using them in reaction. Is it okay now? बात समझ में आ रहा है?
So then, so for example, glucose 6 phosphate has a space. So if we name that as a species, will we need to name that species in double space as well,
Yes,
like in
yes,
double
yes.
quotations?
yes.
Okay.
ओके, सो रिएक्शन कैसे डिफाइन करना है उसमें मैं बता रहा था आपको इवेंचुअली वैसा ही उधर भी डिफाइन करना पड़ेगा व्हेन यू डिफाइन द स्पीशीज इन योर मॉडल. ओके,
Yes, sir.
so the moment I spell out this reaction, and we'll say for example S1 will get converted to S2. Okay, you can have it as reversible. Okay, you can have it as irreversible. रिवर्सिबल किया तो देखो यहाँ पर क्या चेंज हो गया, इट हैज चेंज टू इक्वल. दैट मींस ये इधर के साइड में भी जा सकता है और इधर के साइड में भी जा सकता है, right? S1 get converted, can be converted to S2, and S2 can be converted to S1. For time being we will keep it irreversible. That means S1 has only one direction. S1 can only be converted into S2. Okay,
Okay, sir.
okay, so now देखो यहाँ पर क्या है कि once you have defined this कि S1 will get converted to S2 and it is going to be irreversible reaction, Copasi has detected some rate loss. Okay, and by default it has assign it a rate law of mass action that is irreversible. Okay, so you can also check out key to this particular reaction wherein substrate 1 would be converted to product which is irreversible in nature, what kind of rate laws are applicable to it, what possible rate laws are applicable to it. Okay, so it can be either allosteric inhibitor, it can be catalytic activation, it can be competitive inhibitor, Michaelis-Menten equation, it can follow Hill cooperativity, so far and so forth. Okay, बात समझ में आ रही? Is it okay?
Yes, sir.
Okay. Okay, good. So for our case, I will be considering mass action reversible. Okay. तो ये कौन सा रेट लॉ कंसीडर कर रहे हैं अपन यहाँ पर? वी आर कंसीडरिंग आवर रिएक्शन टू फॉलो द रेट लॉ ऑफ mass action, that is irreversible mass action. Okay. तो what do this rate law signify? Rate law क्या, irreversible rate law क्या signify करता है? What is mass action? Law of mass action क्या कहता है कि the concentration of S2 would be dependent on K and concentration of S1, right? That is in simplest is the rate law of mass action. Okay, तो अपने इसमें पैरामीटर कौन सा है? K1. K1 is the parameter that would determine the conversion of S1 into S2, and this conversion would be dependent on concentration of S1. Okay, substrate क्या है इसका? S1. बात समझ में आ रही है आपको? Is it okay?
Yes, sir.
Okay, देखो अब ये सब डिफाइन किए बरोबर क्या हो गया? All those warning signs are gone. Now things are quite good. Okay, it you know takes some time, so be patient. आपने अगर कर भी दिया and still you see कि यहाँ पर marks है, so maybe click somewhere else and see if that marks go. Okay, it might take some time for it to understand कि सही हुआ है या नहीं. Okay, so I have seen students doing the right thing but they are not patient. अरे जा रहा क्यों नहीं वो? It becomes a kind of don't be impatient in such cases. Okay.
So, but why is the substrate line still highlighted in pink?
नहीं नहीं ये देखो, this is not it is well say for example rate law. ये पिंक का मतलब ये नहीं कि यहाँ पर गड़बड़ है. Okay, it is telling you कि what are the different parameters that are involved in its rate law. Okay, बात समझ में आ रही है आपको?
Yes, sir. Got
Okay,
it.
हाँ. Okay. तो एक दो और चीजें मुझे बतानी है. तो now let us talk about these columns that are, I mean ये column में क्या रहेंगे? Depending on what kind of rate law that you have selected for your reaction, उसमें कितने पैरामीटर्स है, ये चीजें आपको यहाँ पर बताएंगा. ओके, तो अपने रेट लॉ में कितने पैरामीटर्स है? दो ही पैरामीटर्स है. वी हैव गॉट K1, ओके, एंड K1 जो पैरामीटर है and the substrate concentration would determine the way this reaction would behave. Okay, so now और कुछ चीजें हैं जो मैं आपको यहाँ पर बताना चाहूंगा. In general, okay, so now let us talk about this mapping column over here. Okay, this is new for you. Okay, so here we have set the value of mapping to local. Okay, दिख रहा है यहाँ पर? Here it says कि we have mapped the parameter K1 to local. This is how you are supposed to read it. We have a parameter called as K1, okay, and we have mapped this parameter K1 as local. Well, this would mean that the value of K1, that is eventually 0.1, okay, at this stage, the value of K1 will only be confined to reaction R1, जो अपन ने यहाँ पर define किया है. तो ये जो K1 का value 0.1 सिर्फ ये reaction R1 के लिए है. Okay, तो ये चीजें करने का क्या लॉजिक है भाई? वेल से फॉर एग्जांपल, okay, if there is another reaction, Anagha, please हाँ, If there is another reaction which has got, now you are dealing with say some higher level of modeling, जिसमें दो मास एक्शन वाले रिएक्शंस है, तो दोनों में K1 रहेगा, बरोबर है? तो इफ यू मैप इट लोकल, इसका मतलब क्या है, फर्स्ट रिएक्शन का जो K1 रहेगा उसका वैल्यू 0.1 रहेगा, और सेकंड रिएक्शन का जो K1 रहेगा वो अलग रहेगा, दैट वुड बी स्पेसिफिक टू दैट रिएक्शन. Okay, do you get this thing?
So can you repeat once more?
Okay, so local का मतलब क्या है कि whatever the value of this parameter K1 that we have stated over here is 0.1, that would remain local to only reaction 1.
Okay.
Okay, now in your model if you have say 10 reactions, और ये 10 reaction में से दूसरा और कोई reaction है, we'll say for example reaction 9, which is following the same mass action irreversible, तो उसका भी K1 पैरामीटर रहेगा,
बट उसका अलग वैल्यू होगा.
बट अगर लोकल मैप किया तो उसका वैल्यू अलग रह सकता है, एंड इट वुड बी यू हु कैन डिफाइन इट.
गॉट इट.
ओके, बट अगर उसको अब यहाँ पर ग्लोबल इसलिए नहीं आया क्योंकि अपने इसमें एक ही रिएक्शन है, राइट. अगर मल्टीपल रिएक्शन होगा तो यहाँ पर लोकल के साथ में ग्लोबल ही आएगा, ग्लोबल भी आएगा.
और अगर ग्लोबल होता है तो देन से रिएक्शन 1 एंड रिएक्शन
K1
9
would
both
be
will have the same value.
constant throughout your simulation.
Got it. Okay, sir.
Okay,
thank you, sir.
ठीक है, good. So I am deliberately going very slowly, आपको बोर हो रहा होगा, but I want you to understand these things in a very clear way. Okay, टाइम लगा तो चलता है, ठीक है. Okay, so this means that the value of K1 will only be confined to reaction R1. Okay, ये इतना समझ में आ गया?
Yes, sir.
सबकी समझ में आ गया? Okay. Okay. We will for time being keep this value to 0.1. I am just clicking over here just to show you कि आप ये change कर सकते हैं. Okay? यहाँ पर आप जो वैल्यू चाहे वो पैरामीटर की चेंज कर सकते हैं. ओके
Okay, sir.
ओके, ठीक है. तो अगर मैंने इसको वेल से फॉर एग्जांपल माइकलिस मेंटेन का रेट लॉ बताया, तो क्या क्या वैल्यू रहेंगी इसमें?
KMB max.
हाँ, वेरी गुड. इट वुड बी Km रहेगा इसमें, Vmax रहेगा, सबस्ट्रेट कॉन्सेंट्रेशन रहेगा, राइट. सो डिपेंडिंग ऑन व्हाट काइंड ऑफ रेट लॉ यू आर सिलेक्टिंग, यहाँ पर अलग अलग पैरामीटर्स रहेंगे. ओके, इज इट ओके? To summarize for time being, we learn what are species that are involved in our system. Okay, we have learned what are the reactions that are present in our system. ठीक है, where do you check for the rate laws? Okay, and how can you change the values of those rate laws? इतना समझ में आ गया? So this actually kind
Yes,
of
sir.
completes on what is required to set up and build a basic model in Copasi. Okay, so our model is ready now. Okay, is it okay? आगे बढ़ूं क्या मैं?
Yes, sir.
Yes, sir.
Okay, so now well, let us for time being do not talk about things like global quantities. मैंने आपको कंसेप्ट बता दी है ग्लोबल क्वांटिटीज की. ओके, तो कौन से अगर कोई वैल्यू आप ग्लोबल रखना चाहते हैं तो यू कैन सेट इट फ्रॉम ओवर हियर. ओके, you can also set some events. Okay, तो events का मतलब क्या है कि how system or how your model or your compartment would behave if certain conditions are met. Okay, so we'll say for example if you are trying to model division of cell division, तो certain CDK का अगर concentration बढ़ जाता है, right? तो the cell would be triggered to divide, right? तो आप इस तरीके के अगर इवेंट होंगे आपके मॉडल में, तो आप ये इवेंट्स में उसको डिफाइन कर सकते हैं. What would trigger the event? How the trigger would be delayed if it is possible? What are the assignment targets? Exactly क्या होगा? आपके कंपार्टमेंट का वॉल्यूम कम होगा क्या? And all sort of things are to be done over here. Events में. For time being, well, let us not go over there. Okay. Well, parameter overview में, हाँ, this is very important. Okay. So parameter overview would tell you a brief idea of your model. Okay, so parameter overview gives you an overall idea of a model, like the initial time. Initial time क्या है अपने मॉडल का? 0 है. Why? Because we are starting it from scratch. What is the initial compartment size? Okay, we have kept it to fix. What are the initial species? Okay, what are the kinetic parameters? Reactions की K1 है, उसको fix रखा है, उसका value 0.1 है. Okay, now what you can do is that you can save this to a file. Okay, आप इसे सेव कर सकते हैं. I am just clicking it over here. Okay, and you can save it. दिख रहा है क्या? Save as का कॉलम दिखा आपको?
Yes, sir.
Okay, so you can save it and use this tab separated value. You can import it further, right? So this is also possible. Now let us move on to next menu, that is mathematical equation. Okay, तो यहाँ पर देखो, you have got this mathematical equations. Okay, so mathematical equation में differential equation है. So based on whatever model you have built so far, okay, so it shows that there are two differential equations that this Copasi has derived from whatever model that you have built so far. Okay, so it has two sub menus, जैसे कि यहाँ पर दिख रहा है आपको, differential equation and matrices. अब एक एक करके मैं आपको बताऊंगा कि इसमें क्या क्या होता है. Okay, now if you click on this differential equation, right hand side में आपको दो differential equation दिखेंगे. Okay, so what do these differential equation says? Okay, so इसमें से जो दो differential equation है, it correspond to d by dt, यानी क्या, rate of change of this substrate 1, okay, or with respect to the cell size. And देखो यहाँ पर आपको you can see there is a minus sign over here. Okay, और दूसरा क्या है, S2 जहाँ पर plus sign है, this differential equation, right? So it would indicate that this differential equation is responsible for defining rate of consumption of this S1, and this S2 would determine rate of formation of S2. Okay, so this equation says that the rate of equation, that is d by dt of species S1, okay, that is concentration of S1 of reaction R1, right? Here we have two differential equations, जैसे कि मैंने बता दिया. Okay, now I sincerely hope that you have attended the previous lectures that Dr. Vivek has conducted. Okay, and I hope that you are well versed with how do we represent our reactions in differential equations. What are the stoichiometric matrices that are possible for these differential equations? Okay, वो सारे यहाँ पर रहेंगे. Okay, this is the stoichiometry matrix. ये हुआ ना theory में?
Yes, sir.
हाँ, how do you
हाँ, sir,
generate
हो
the
गया.
stoichiometric matrix, right? इसके अलावा भी matrices है, जो जैकोबियन कंप्लीट मैट्रिसेस है, जैकोबियन रिड्यूस मैट्रिसेस है, जो हो सकता है कि आपको आगे बताया जाएगा. बट आई एम डैम श्योर कि स्टाइचियोमेट्री मैट्रिक्स आपको जरूर पता रहना चाहिए. अभी
हाँ सर, पर वो रिड्यूस
sir.
वाला like
वो बाद में देखेंगे उसका क्या
ओके
है.
ओके हाँ हाँ ओके.
ठीक है, तो ये डिफरेंशियल इक्वेशन है, उससे रिलेटेड ये स्टाइचियोमेट्री मैट्रिसेस है. ओके, The next thing that you have is diagram. This is also one of the interesting feature over here. Okay, so what you can do, जैसे आपने सेल डिजाइनर यूज किया होगा, किया क्या?
Yes, sir.
ओके तो उसमें ऑलरेडी आप बना सकते हैं, यहाँ पर आपको ये बनाना पड़ता है, ठीक है. सो इफ यू गो टू दिस डायग्राम, यहाँ पर न्यू है, नीचे मैंने न्यू के ऊपर में क्लिक किया था, इट वुड टेल यू कि व्हाट डायग्राम डू यू नीड टू मेक. ओके, आई नीड टू मेक डायग्राम फॉर यू नो a compartment called as cell. Then what reactions do you want to include in your diagram? Reaction 1. Okay, and all you need to do is to click next. अब रिएक्शन में अगर मल्टीपल ये हो गए तो यू कैन ऑल्सो सिलेक्ट हाउ मेनी स्पीशीज दैट आर गोइंग टू बी देयर इन योर डायग्राम. ये सिलेक्ट करने का यू नीड टू टेक फ्रॉम स्पीशीज साइड टू द साइड स्पीशीज साइड. ओके, ये इधर आ गए एंड फिनिश करते ही देखो एक नए इसमें डायग्राम आ जाता है. नाउ इट शोज यू कि दिस स्पीशीज S1 would be converted to S2, right? Then you can change the layout or you can render the information. जैसे gradient आप change कर सकते हैं. You can play on with these different stuff. Okay, you can save it as you can export it as an image. Okay, you can view it to full screen. I would let you know, let you guys to explore these things, but this is how you create representation for your metabolic model. Okay, बात समझ में आ गया बेटा? Is it okay?
Yes, sir. Yeah.
Okay, now I am closing this thing. Okay. Okay, so here is what to conclude, this is how you create an overall model. Okay, बात समझ में आ रही? Where this is how you infer the differential equation, right? Is it okay? Now let us talk about task. तो ये टास्क में तो मॉडल तो तैयार हो गया अपना. Is it okay? Now let me close it. ये भी बंद कर देता हूँ, ये भी बंद कर. So once the model is ready, what task you can do, right? तो इसमें आप बहुत सारी चीजें कर सकते हैं. You can perform steady state, you can perform stoichiometric analysis, you can perform time course simulation, you can perform metabolic control analysis. Okay, तो इसमें से today we would be performing time course analysis. Okay, तो अगर आप ये करते हैं अगर आप ये टाइम कोर्स के ऊपर में क्लिक करते हैं तो यहाँ पर देखो on the right hand side you would get all the values that would define or that would tell how the time course simulation is needed to be performed. Okay, so what it says for time being is like I have one reaction, okay, with two species. If I start with concentration of species S1 and S2, okay, S1 as 0 and S1 का concentration क्या सेट किया था अपन ने? S1 का concentration क्या सेट किया था अपन ने? 1. और S2 का क्या किया था? 0. And let the reaction happen over time. Then how does the concentration of S1 and S2 would change over time? Okay, that is what this time course simulation does. Okay, तो अपना मॉडल में क्या है? We have got two species S1 and S2 that would participate in a reaction named R1. Okay, and if I let this reaction to happen over time, how would it change the concentration of S1 and S2? That is what you would gain when you perform this time course simulation. Okay, so there are a lot of things that can be done जो उसके right hand side में आपको दिखेंगे. Okay, there are a lot of options that would define how your simulation would run. तो उसमें से कुछ basic चीजें मैं आपको अभी यहाँ पर बताने वाला हूँ. Okay, by default we have simulation of 1 second over here, right? We have simulation of 1 second with interval of 100 and interval size of 0.01. Okay, तो इसमें आपका उधर एमडी सिमुलेशन हुआ है क्या? टाइम स्टेप एंड नम स्टेप, वो सारी चीजें, these are analogous to that thing.
Yeah, sir.
Okay, so you can define it over here, and the best portion over here is that जैसे ही आप ये चेंज करते हैं 1 सेकंड टू वेल से फॉर एग्जांपल 10 सेकंड किया, तो उसके हिसाब से इंटरवल साइज भी चेंज होता है, देखा? So you don't have to worry about ki how do I define the interval size, how do I define the intervals. It would take care of everything for these things. Huh? This is wonderful, right? So what I have done is that change the duration of our simulation for 10 nanosecond, sorry 10 second, and eventually उसके साथ में इसका interval 100 का रहेगा. Okay, so for time being, well, first let us simulate our reaction for 10 seconds. Okay, with an interval size of 0.1 with 100 intervals. Okay, so इसके अलावा now since we are using, do you remember अपन ने मेथड कौन सा यूज किया था? डिटर्मिनिस्टिक मेथड यूज किया था, राइट? तो उसके भी अलग अलग एल्गोरिदम्स अवेलेबल है, राइट? तो यू कैन चेक ओवर हियर. तो आप डिटर्मिनिस्टिक भी यूज कर सकते हैं, स्टोकेस्टिक भी यूज कर सकते हैं, और बहुत सारे मेथड भी है यहाँ पर, हाइब्रिड मेथड भी यूज कर सकते हैं. ओके, सो फॉर टाइम बीइंग लेट अस यूज द डिफॉल्ट मेथड, डिफॉल्ट एल्गोरिदम. तो उसके पैरामीटर्स भी आप यहाँ से चेंज कर सकते हैं इफ यू विश, राइट? So well, let us not go in more details of this thing. For time being, well, let us use the deterministic method called as LSODA for performing the time course simulation of our reaction. Okay, is it okay? Okay,
Yes, sir.
ठीक है. तो अभी एक और चीज आपको करनी है. In order to do these things, you should also know the output specification. This is also important to be defined. Okay, so if you click on output specification, ये आपको बताएगा कि आपका आउटपुट कैसा रहेगा, so that you can analyze the data. Okay, तो यहाँ पर एक आउटपुट असिस्टेंट भी है. ओके, तो ये आउटपुट असिस्टेंट आपको क्या बताएगा कि व्हाट काइंड ऑफ plots that you can create, what kind of reports that you can create. So for time being, we would select this constant first plot that we would plot. ये क्या बताता है कि we are going to plot the variable species concentration, variable compartment volumes, and variable global quantity versus time. Okay, is it okay? बात समझ में आ रही? तो यहाँ पर आप आउटपुट असिस्टेंट से आपको एग्जैक्टली क्या क्या आपके आउटपुट में चाहिए वो चीजें आप डिफाइन कर सकते हैं. ओके, सो वी हैव सिलेक्टेड दिस वन, कंसंट्रेशन, वॉल्यूम्स एंड ग्लोबल क्वांटिटी वाला. ओके, सो इफ यू यूज दिस क्रिएट, ओके, इट हैज क्रिएटेड. देखो यहाँ पर 0 था, अभी एक प्लॉट तैयार होगा यहाँ पर 1, राइट? एंड दिस प्लॉट वुड प्लॉट व्हाट? कंसंट्रेशन, वॉल्यूम्स एंड ग्लोबल क्वांटिटीज. बात समझ में आ रही? Okay, now all you need to do is to click this run. Okay, and here are the results. Congratulations, you have got your first results. So now let us analyze these results. Analysis से क्या समझ में आएगा? It is, the results are quite clear. Okay, क्या बताते हैं? This is S1. We have started with 1, बरोबर है? उसका initial concentration क्या था? 1. As the reaction has proceeded further, eventually the concentration of reaction, the concentration of your species 1 would decrease. बरोबर है? Why? Because it is being used up in production of species S2 that has begin with 0 and eventually it is growing up. बरोबर है?
Yes, sir.
So this is how the things are supposed to be interpreted. Okay, ये ग्राफ क्या बताता है? समझ में आ गया क्या आपको? ये ग्राफ बताता है कि जैसे ये रिएक्शन फ्रॉम सेकंड 0 से शुरू हुआ एंड इट एंडेड अप एट 10th सेकंड, the initial concentration of species 1, which is the reactant of our system, was used up eventually slowly, and its concentration is going on decreasing. And as the reaction proceeded from 0 to 1, okay, from 0 to 1, sorry, from 0 to 10, subsequently or linearly, the reactant is forming, sorry, the product is forming, इसलिए उसका concentration बढ़ रहा है. Okay, is it okay?
Sir, I don't see any graph.
Okay, okay,
Sir,
okay.
are you showing any graph?
Yes,
Where-
yes,
Yeah.
yes. Maybe I have to share it. Okay, sorry, huh? Okay. Now can you see the graph?
Yes.
Okay. हाँ बताओ रे, ऐसा नहीं दिख रहा है. हाँ तो ये दिस इज द रिजल्टिंग प्लॉट. ओके, तो ये प्लॉट में क्या दिख रहा है आपको? द पिंक कलर और रेड कलर लाइन क्या इंडिकेट कर रही? It
S1.
it
S1
indicates the
substrate.
concentration of substrate that is being depleted with time.
Yes.
And S2 जो blue color का plot है, that indicates the
Increased.
increased concentration of the product. And the crossover here, the point at say some 7 indicates that
S1 equal to S2.
the concentration of substrate and concentration of product were exactly equal.
Yeah.
Right?
Yes, sir.
Now we will do one more interesting thing. Well, this is expected, right? This is expected since we started
Yes.
with the concentration of S1 at 1 and S2 at 0. We see steady depletion of S1 and steady increase of S2 as reaction proceeds.
Yes.
Right?
Yes, sir.
Okay, okay. Now अभी एक और चीज करते हैं. Well, now let us try to increase the duration of our simulation. Okay, now I will close this. Now do you still see my Copasi window?
Yes, sir. We can see.
Yes, sir.
Okay, तो अभी क्या करते हैं, अब पहले अपने कितने सेकंड के लिए सिमुलेट किया था इसे?
10.
10. तो इसको अभी क्या करेंगे अपन? We would simulate it for 100 seconds now. Okay, तो देखो 100 सेकंड करते ही इसका इंटरवल इसने अपने आप बढ़ा दिया और इसका इंटरवल साइज और कम कर दिया. इंटरवल साइज भी बढ़ा दिया, वैसे फॉर एग्जांपल. Okay, so it is very intuitive and it makes
Sir,
your
the
life
interval,
easier.
the number of intervals increased because the interval size is kept constant, right?
Yes. Okay, now again we will run our simulation. Now I will again share this thing. I would have to share so that you can see.
So we can see it, yeah.
We can see it, sir.
You can see this?
Yeah.
ये नया वाला प्लॉट दिख रहा है आप लोगों को?
हाँ.
Yes,
We can
sir.
see.
Okay, now here is something interesting. क्या किया अपन ने? We have increased the time of simulation, right? From पहले जो सिर्फ 10 तक था, दिख रहा है यहाँ पर 10 तक था, अभी अपन ने क्या किया, उसका टाइम 100 कर दिया.
Yes.
Okay, तो इससे क्या हुआ? What do you see? What difference do you see?
Since the number of intervals increase, so I think the plot just smoothened out further, and this is a more realistic scenario basically. So
देखो इसको कैसे इंटरप्रेट करना मैं आपको बताता हूँ. Okay, you can see that with due course of simulation time, initially when the reaction began, okay, there was subsequent change in your system, okay, in which the concentration of this species 1 eventually started to decrease, and at say some point when it reaches about 70 second, after 70 second, there is there is no evident change in the concentration of S1, बरोबर है? कुछ कुछ ड्रास्टिक चेंजेस हो रहे हैं क्या ये concentration of S1 में?
No, sir.
Same is the case with the product formation, that is the concentration of S2. इसमें भी कुछ drastic changes हो रहे हैं क्या?
No, sir. The saturation point is reached.
That means the system has reached steady state.
Yes.
बराबर है? So if you perform this time course simulation of longer enough time scale, you can check out if the system reaches the steady state. बरोबर है?
Yes, sir.
तो steady state reach हुआ ऐसा कब कहते हैं अपन?
When their concentrations do not change
Do not
after that
change.
point
Rate of formation equates rate of
depletion.
depletion. देखो, it has reached a kind of convergence. इसका मतलब क्या? इसमें ज्यादा changes नहीं हो रहे. That means it has reached steady state.
Yes.
Right? बात समझ में आ रही?
So basically,
Yes,
this
sir.
time there will be no more change in concentration.
There is no more subsequent. अगर आप इसको 1000 करेंगे तो भी ज्यादा change नहीं होगा.
हाँ मतलब हाँ, so it won't matter.
That so that you can say कि your system has now reached steady state. देखो normal biological system में भी ऐसा ही होता है. Your system should reach steady state.
Yes.
Right?
Yes.
Then and there you can say कि आपने एक you know एक acceptable model तैयार किया है.
So, sir, if in case you ask us when did it reach steady state, so do we answer according to this plot? So we see a break at around 70, right?
70.
Or somewhere around like 60, between 65 70. So do we answer it like that, that at 69 or
Around
70?
60 or 70 the system reaches steady state. This is how you are supposed to do.
Okay.
Okay. तो देखो biological systems भी इसी तरीके के होते हैं. Okay. जिसमें initially आप कोई perturbation करते हैं तो things would change but eventually your system would get used to it. Okay. And you would eventually obtain the steady state. You would eventually reach homeostasis. Okay. बात समझ में आ रही है बेटा?
Yes, sir.
Okay, so
Yes, sir.
whenever you create any biological system या biological model आप जब भी तैयार करते हैं तो make sure that you perform time course simulation or steady state analysis in order to see कि भाई आपका जो system है वो steady state reach करता है, it converges at some point, right? Then you can proceed for further analysis. आपको जाना चाहिए आगे. Okay. Is it clear? बात समझ में आ रही है?
Yes, sir.
So this is all I have for you guys today. Okay. And now what you are supposed to do is that you guys try to simulate this model. I will share this video to you on your drive. Okay. You check out this video, do these things take your own time. You have got entire entire week to do that. And maybe what I would ask you to do is that try to change different type of kinetic parameters. Okay. उसके रेट लॉस चेंज करके देखो. अभी ये काय के लिए बनाया अपन ने? इसके लिए ये रिएक्शन के लिए रेट लॉ कौन सा यूज किया था अपन ने?
Mass action.
मास एक्शन. तो माइकलिस मेंटेन वाला यूज करके देखो.
Okay, sir.
Right? Try to make it reversible and see how things happen. Right?
So,
Yes, sir.
तो आप यहाँ से इसको सेव भी कर सकते हैं. Right? Save image, save data. Okay, this is also interesting. अगर आप इसका सेव डेटा करते हैं तो टेक्स्ट वैल्यू में आपका डेटा सेव होगा. Okay, and you can plot it using your favorite Microsoft Word, Microsoft Excel or any other spreadsheet application. Okay, तो आप यहाँ से भी कर सकते हैं. You can also see that there are reports and templates that have been created. Okay, there are 11 of those. Okay, तो ये चीजें करो. Try exploring these things now. Right? समझ में आ गया सब कुछ?
Yes, sir.
Yes.
Okay.
So, I had one doubt regarding the second assignment.
Uh-huh.
So, for modeling that, like for getting those equations, when we incorporate the inhibitors, do we just give values of k or do we have to figure it out?
हाँ देखो ना, हो सकता है लिटरेचर में आपको कहीं पर मिल जाए.
Because we looked in databases and they give the concentration of the inhibitor they've given. They don't mention ki anywhere. So then I was confused as to how we take that value.
ओके तो ये ऐसी चीजों के बारे में ये जो आपको आया है ना, ये पैरामीटर एस्टिमेशन नाम की एक कंसेप्ट है यहाँ पर.
Okay.
तो आप पैरामीटर एस्टिमेशन से भी वो चीज ये कर सकते हैं. पैरामीटर ऑप्टिमाइजेशन, पैरामीटर स्कैन, पैरामीटर एस्टिमेशन, these things can be done from over here as well. तो देखो, it is all up to you to read that user manual first.
Okay.
ठीक है, a lot of things can be done over here, right? देखो मैं आपको हर एक चीज तो नहीं बता सकता बेटा. Okay, so I would now leave you to explore all these things. Okay.
Sure, sir.
तो मे बी अगली बार मैं एक और एग्जांपल लेकर आऊंगा जिसमें अपन कुछ चीजें पढ़ेंगे, राइट, सिमिलर स्टफ. तो ये इतना समझ में आ गया, हाउ टू सेट अप अ मॉडल, how to make sure everything is right, and how do you proceed for time course simulation, how do we plot it, how do we analyze it. if you want to make perturbation, आपको कहाँ पर कौन सी चीजें कब चेंज करनी है, ये समझ में आया है. इनिशियल कॉन्सेंट्रेशन मैंने अगर कहा कि अपने को चेंज करना है, तो आपको समझ में आना चाहिए कि वी नीड टू गो इन दैट मॉडल सेक्शन और रिएक्टेंट के वो टेबल में, वो कॉलम में मुझे चेंज करना पड़ेगा, राइट. so it might take some time for you to digest this thing. so my best advice is to इसको बहुत बार आप क्या करो, perform these experimentations one after another. Okay, and just play around with these things, see how things happen. Huh?
Sure, sir.
Okay, ठीक है. तो इसका मैं क्या करता हूँ, I will file save this as time course steady state मैंने सेव करके ही रखा है, right? So I will share this model with you. Okay, ये कहाँ पर शेयर करूंगा मैं, आपके आपके
MESP
गूगल
folder.
ड्राइव के ऊपर मैं शेयर करूंगा, so that if something went wrong you can check out this model as well. Okay, बात समझ में आ रही? ठीक है.
Yes, sir.
Are there any doubts now? Are there any doubts now?
No.
नहीं? Okay, so I am stopping.